Hi, I'm Rokib Hasan, a PhD candidate in molecular bioscience PhD program here at AState, and my advisor is Dr. Fabricio Medina-Bolivar. Today, I'm presenting my research on finding the anti-inflammatory mechanism of Arachidin-3. Around 40% of the hospitalized coronavirus patients develop ARDS, which is caused by lung inflammation due to the virus infection. Because of the inflammation, fluid and immune cells are going inside the lung alveoli, resulting interruption of gas exchange, which causing breathing problem, and patients needed ventilation to breathe. Unfortunately, almost 50% of those ARDS patients are dying based on the current research. So lung inflammation is causing death in this case. Chronic inflammation can also cause and help to progress many other diseases like heart disease, cancer, Alzheimer's, etc. Even though there are few anti-inflammatory drugs available, scientists are constantly looking for more targeted, improved, and less side effect containing drug candidates. If you look closely among the anti-inflammatory drugs here, the latest one is Celecoxib, which was invented in 1995, more than 2 decades ago. Resveratrol, which initially found in red wine and later also found in grape, peanut, and blueberry, showed wide range of health benefits, including anti-inflammatory and anti-cancer activities. However, because of faster metabolism, it has limited bioavailability. Arachidins, which found in peanut, had similar backbone of resveratrol with an extra 5-carbon chain called prenyl group. Collaborative research from our lab found that because of prenyl group, arachidins are potentially more bioavailable, can inhibit virus replication, and can also bind with the cannabinoid receptor. Some literature is also suggesting anti-inflammatory effect of arachidin. However, the exact molecular mechanism of anti-inflammatory effect of arachidin yet to discover. Our lab uses peanut hair root system to produce these arachidins. In this project, we will elicit, extract, and purify the arachidin-3 from the peanut hair root medium and check its anti-inflammatory effect in cell line and rats. Overall goal is to dissect the potential anti-inflammatory role of the arachidin-3, and the hypothesis is that arachidin-3 has better anti-inflammatory activity than resveratrol. After elicitation and extraction of the peanut hair root culture medium, Arachidin-3 was purified using HPLC method, which later has been used to treat the cell lines. Cytotoxicity and cell viability has been checked using CellTox screen cytotoxicity and real-time glow empty cell viability assay, respectively. Cytotoxicity assay results showing that 5 and 2.5 micromolar concentration is not toxic compared to the vehicle control 0 micromolar, which is the orange line. Similar to cytotoxicity, there is no effect of cell viability in lower concentration of arachidin-3 treatment compared to the control. Later, LPS-induced inflammatory cells were treated with arachidin-3 and found that arachidin-3 significantly reduces the pro-inflammatory cytokine IL-6 production compared to the control. NF-kappa beta signaling pathway plays crucial role in inflammation, and many drugs are specifically targeted for this pathway. Inhibition of any major molecule in this pathway can reduce inflammation, specifically inhibiting the translocation of the REL-A, which is also known as the P65, can hamper the pro-inflammatory cytokine production. So we did some computational analysis which suggesting that arachidin-3 has stronger binding affinity for P65 and IKK. To test that, we have used immunofluorescence-based imaging technique and found that arachidin-3 inhibits the translocation of NF-kappa beta P65 complex and also the HMGB1. These results suggesting that arachidin-3 acts as an anti-inflammatory compound by regulating the NF-kappa beta signaling pathway. We have also conducted proteomics from UMS Proteomics core facility, where LPS-induced inflammatory cells were treated with arachidins and resveratrol as a positive control. And an animal pilot study on rats have been conducted in collaboration with Dr. Rajagopalan from NYIT to have better understanding of anti-inflammatory functions of arachidins. These experiments are ongoing and data need to be further analyzed. In conclusion, we have found that lower concentration of arachidin-3 is safe, and it functions as an anti-inflammatory agent by inhibiting the RelA complex translocation into the nucleus, which eventually reduces the production of IL-6 and reducing the inflammatory responses. For further experiment, we will use Western blotting and other molecular techniques to check how arachidin-3 affecting other key proteins such as IKK and I-kappa beta in this pathway, and further animal testing will be done for better understanding. Here is the references that I have used. And I want to thank everyone for the research support, including the funding agencies. Thank you so much.